Welcome back to the deep dive. Today, we are standing at the foot of a mountain.
A mountain made of money, organic chemistry, and, well, a whole lot of bureaucracy.
And we're looking up at what is arguably one of the most complex, expensive, and tightly regulated processes in the modern world. We are talking about the birth of a medicine.
It's a journey that I think most of us take completely for granted. You wake up, you feel a headache coming on, you walk to the cabinet, and you swallow a white pill.
Right.
20 minutes later, the pain is gone. But the story of how that molecule, that specific chemical, ended up in that bottle, it's an epic. I mean, it's a story of failure, of massive financial risk, and just occasionally of scientific brilliance.
And to guide us through this epic, we aren't just looking at, a quick summary online. We are synthesizing two of the absolute Bibles of medical education. We have Ketzing and Trevor's Basic and Clinical Pharmacology, and we have the heavyweight champion, Goodman and Gilman's The Pharmacological Basis of Therapeutics.
And you really need both. Think of these two books as the architecture of our understanding today. Ketzing is going to be our scaffold.
The scaffold.
Yeah, it provides the clarity, the organization, the clinical structure. It tells us where all the pieces go. It gives us the framework.
And Goodman and Gilman.
That's the deep foundation. That gives us the mechanistic depth, the molecular reasoning, the systems level insight. It explains why things happen the way they do, right down to the level of the atom.
So we're combining the scaffold and the foundation to build a kind of conceptual map of drug invention. And I want to be really clear right up front. This deep dive is an interpretive synthesis. We are turning on the lights in the room. But if you are a medical student or a clinician listening to this, you absolutely still need to read the chapters.
Oh, absolutely. Consider this the pre-read that makes the actual reading, well, let's say, a lot more bearable. It gives you the big picture before you dive into the details.
Fair enough. Now, before we get into the chemistry and the trials, let's set the coordinates. What's our mission here? What are the key takeaways, the pillars we need to build for you?
We have 5 main goals for this deep dive. First, we need to understand the drug journey as a repeatable framework, from identifying a target in a cell all the way to a product on a pharmacy shelf. It's a process.
A very long process.
A very long process. Second, we have to tackle what's called the translation problem. This is the, frankly, heartbreaking reality that what works in a mouse in a lab rarely works in a human.
And we're going to spend some real time on that because that failure rate is just staggering.
It is. It really is. Third, we're going to look at regulation, not as, boring paperwork, but as a safety infrastructure that is quite literally written in blood.
What do you mean by that?
Every major regulation we're going to discuss today is basically a tombstone for a past tragedy. It's a reaction to people getting hurt or killed.
Wow. Okay. What's number 4?
Fourth, we have to face the industry realities, the economics, the patents, the so-called Me Too drugs, the stuff that makes people uncomfortable.
The constant tension between scientific idealism and the need to turn a profit.
Exactly. And finally, number 5, we need to adopt the clinician's lens. We need you to stop viewing drugs as just isolated chemicals in a vacuum and start seeing them as powerful interventions in complex, dynamic living systems.
Okay, that's a fantastic roadmap. So let's jump right in with the distinction that appears early in Goodman and Gilman. It challenges the way we even talk about this whole industry. We usually hear the phrase drug discovery.
Right, sounds romantic.
It does, like you're Indiana Jones, finding a lost artifact in a jungle temple.
But the text argues pretty forcefully that we should be calling it drug invention.
What's the functional difference there?
Yeah.
Invention versus discovery.
It's a profound shift in mindset. Discovery implies that the perfect drug is already out there, maybe hiding under a rock or in a rainforest leaf, and we just have to stumble upon it. And to be fair, historically, that was kind of true.
We found things.
We did. We found morphine in poppies. We found digitalis in the foxglove plant. It was often pure serendipity, a happy accident.
We were tripping over cures.
We were, in a way, but we aren't just stumbling around in the forest anymore. Today, useful drugs are rarely just found. They are sculpted, they are engineered. We identify a biological locker receptor, an enzyme, and then we painstakingly, rationally carve a key to fit it.
So it's deliberate.
It's a process of deliberate creation. We optimize its properties, we change its shape atom by atom, we invent it.
And that invention process is grueling. I think it's important we establish the scale here. It's not like you invent a drug on Monday and it's in the pharmacy by Friday.
Oh, not even close. Far from it. You're looking at maybe 10,000 initial chemical candidates to get one single approved drug. You're looking at an investment of over $1 billion, often closer to 2 or 3 billion by some estimates.
$1 billion for one medicine.
And you're looking at a timeline that consumes entire careers, 10, 12, 15 years from lab bench to bedside. It is a high-attrition, high-stakes game where failure is the default state. Most things do not work.
So if we are inventing these things, let's start at the absolute beginning, at the atomic level. What exactly are we inventing? Katsung gives us the nuts and bolts of what a drug actually is, its size, its shape, the bonds it makes. So let's start there in section 1. The nature of drugs. Does size actually matter in pharmacology?
It matters immensely. I mean, it's one of the most fundamental properties. We measure drugs by their molecular weight, or MW, and there is a definite Goldilocks zone for most drugs.
Not too big, not too small.
Precisely. The sweet spot is generally between a molecular weight of 100 and 1,000 Daltons.
Okay, let's unpack that. Why is there a lower limit? Why can't we have a tiny little nano drug with a molecular weight of, say, 10?
One word: specificity. This is probably the most crucial concept in all of pharmacology. Think of a drug as a key and the receptor in your body as a very specific lock.
Okay.
If your key is just a tiny, simple pin with no complex grooves or shape, it's going to fit into thousands of different locks. It will unlock your front door, but also your neighbor's car and the bank vault and the bathroom stall.
Which, translated to the human body, means side effects. Just rampant side effects.
Massive, unpredictable side effects. A molecule that is too small lacks the structural complexity to be unique. It will bind to receptors all over the body, not just the one you want to target. You need a certain amount of size, usually an MW of at least 100, to have enough grooves and ridges and different functional groups on the molecule to fit only the specific receptor you're targeting.
It needs to be complex enough to hold a unique code, essentially.
Exactly. Uniqueness requires complexity, and complexity requires a minimum size.
That makes perfect sense. But then there's the upper limit, 1,000 milliW. Why stop there? Why not make a super complex giant molecule?
Because of movement, permeation. Pharmacology isn't just about the drug hitting the target. It's about the drug getting to the target in the first place.
Right, it has to travel.
It has to travel. And large molecules, giant proteins, for example, don't diffuse well. They're like trying to drag a sofa through a crowded subway car. They can't move between the body's compartments easily. They can't cross the blood-brain barrier. They certainly can't get absorbed from your gut into your bloodstream efficiently.
So if a drug is huge, you can't just take it as a pill. It won't get absorbed.
Usually no. That's why things like insulin, which is a protein, or these newer monoclonal antibodies like Erinumab for migraine, they all have to be injected. You have to place them directly into the compartment where they need to work, which is usually the vascular system, because they are simply too big to wander around the body on their own. They can't cross the gut wall.
So the ideal drug is big enough to be specific, but small enough to be mobile.
That's the balance you're always trying to strike.
Okay, so once it gets there, it has to stick. It has to interact. Let's talk about bonding. Goodman and Gilman breaks this down into 3 main types: covalent, electrostatic, and hydrophobic.
Right. And the difference really comes down to the level of commitment the drug has to the receptor. It's like different kinds of relationships.
How committed are we talking?
Well, the strongest bond is the covalent bond. This is a marriage. It is strong, it is stable, and it's often irreversible. The classic example that both texts highlight is aspirin.
Aspirin is aggressive.
In a chemical sense, absolutely. Aspirin binds to an enzyme called cyclooxygenase, or COX, in your platelets. It doesn't just stick to it, it forms a covalent bond. It literally fuses with the enzyme. It permanently breaks it.
So the effect doesn't stop when the aspirin leaves your blood.
No, and that's the fascinating and clinically vital part. The aspirin molecule itself might be cleared from your bloodstream in 15 or 20 minutes. But that platelet, it's done. It can never produce thromboxane and help form a clot again. The effect of that single dose of aspirin lasts for the entire lifespan of that platelet, which is about 7 to 10 days, until your bone marrow makes new ones.
That explains why surgeons tell you to stop taking aspirin a week or more before surgery. It's not about the drug currently in your system. It's about the entire population of cellular machinery you've permanently disabled.
Exactly. If you cut someone open who took aspirin 3 days ago, their blood is still thinner. They will still bleed more than normal, even though there's no aspirin in their plasma. But most drugs aren't like that. Most use electrostatic bonds.
Okay, what are those?
These are much weaker. They are more like a magnet on a fridge or a sticky note. It's a casual dating relationship.
Reversible.
Totally reversible. The drug floats in, sticks to the receptor because of charge attraction, positive to negative. It activates it or blocks it for a moment, and then it floats away. As the concentration of the drug in the blood drops, the drug unsticks and the effect wears off.
And that's safer, I'd imagine.
For most therapies, it's much, much safer because if you overdose or if you have a bad reaction, you can just stop the drug and it washes out. You're not stuck with a permanent effect for 10 days.
So that's size and bonding.
Yeah.
The third element from Katzung is shape. And this brings us to one of the most mind-bending concepts in chemistry, chirality.
Handedness.
Right. The idea that a molecule can have a left-handed version and a right-handed version, like mirror images.
It's absolutely crucial. These are called enantiomers. And just like your left hand acts very differently than your right hand, I mean, try putting your left hand into a right-handed glove. A left-handed drug molecule acts differently in the body because biological receptors are incredibly precise stereochemical environments. They are the glove. They're built to fit one hand, not the other.
And this isn't just some esoteric chemistry trivia point. It has real life or death safety implications.
Huge implications. Sometimes the other hand is just inactive. It's dead weight. It doesn't do anything. But sometimes the other hand is actively toxic. The classic tragic example is thalidomide. One enantiomer might cure your morning sickness, and its exact mirror image might cause catastrophic birth defects.
You're alluding to the thalidomide tragedy there, of course.
I am. Thalidomide was sold as what we call a racemic mixture. It contained both the left and right-handed versions of the molecule. One was a sedative, the other was a teratogen that caused phocomelia.
And we will definitely get to the full horror of that story in the regulation section. But from a design perspective, this means modern drug invention has to involve rational design. You have to sculpt the molecule to be purely the active shape, the correct hand, eliminating the useless or, worse, the dangerous mirror image.
Exactly. We don't just throw the whole mixture in anymore. We purify the desired enantiomer. This is a key part of moving from accidental discovery to intentional, precise invention.
Okay, so we have our blueprint. We know what we're trying to build. A molecule the right size, making the right kind of bonds, with the right three-dimensional shape. So now, let's move to section 2. The invention process itself. How do we actually do this today? You mentioned we don't rely on serendipity and happy accidents anymore.
For the most part, no. We rely on a process called high-throughput screening, or HTS. Imagine you have a chemical library owned by a pharmaceutical company. This isn't books. It's millions and millions of unique chemical compounds stored in tiny wells on plates.
And you test them one by one.
In the old days, a chemist would, and it would take a lifetime. Now, we use robotics and computers to basically roll these millions of chemicals over a purified target protein, the lock we're trying to pick, to see if any of them stick.
But wait, before you can roll the chemicals, you need a target. You need the lock. And Goodman and Gilman list some absolutely crucial questions you have to ask before you even start up the robots. The first one is the target druggable?
It sounds like a made-up word, doesn't it? Druggable. But it's a rigorous and often painful technical assessment, Damien. It means, does this biological protein, this enzyme or receptor, have a well-defined binding site, a pocket, a cleft, a groove that a small organic molecule can actually fit into with high affinity and specificity?
Because some proteins are just, big, smooth, slippery spheres there's nowhere to grab on?
Essentially, yes. Or their active sites are buried too deep inside the protein's folded structure. Or the interaction surface is huge and flat, like two flat plates touching. A small drug can't disrupt that kind of large-scale protein interaction. If a target isn't druggable, you can have the best theory in the world about how it causes disease, but you simply can't make a traditional pill for it.
So you have to abandon that whole line of research.
Or you have to invent a completely different modality. You switch from trying to make a small molecule to trying to make a monoclonal antibody or a gene therapy, which are different beasts entirely.
Okay, so let's say it is druggable. The second question from the text is it validated?
This is the billion-dollar gamble. Validation means asking, if we actually succeed in hitting this target, does it actually change the course of the disease in a meaningful way?
You would think they would know that for sure before they started spending hundreds of millions of dollars.
You would think so. But biology is incredibly messy and complex. Often, we rely on animal models. We knock out a gene in a mouse, and the mouse's version of the disease gets better. So we say, great. The target is validated.
But then
we spend 10 years and $1 billion making the perfect drug. We give it to a human, and absolutely nothing happens.
Why? What's the disconnect?
Redundancy. Humans have incredible biological redundancy. We have backup pathways. You block pathway A, and the human body just calmly reroutes all the biological traffic through pathway B. The mouse didn't have pathway B, or it wasn't as important. This retrospective failure of target validation, finding out you picked the wrong target after you've already built the drug, is a huge cause of late-stage industry attrition.
So you can do everything right scientifically and still fail because you started with a flawed premise.
Precisely. It's why so many promising drugs fail in the big, expensive human trials.
And then there's the third question, the one that makes people uncomfortable. Is it economically viable?
It's the elephant in the room of modern medicine. Drug invention is overwhelmingly driven by private capital. Investors need a return. So if you invent a perfect cure for a very rare disease that affects only 50 people worldwide, or a disease that primarily affects people in developing nations who cannot pay thousands of dollars for it, the investor-owned pharmaceutical model breaks down.
There's no market.
There's no viable market to recoup that billion-dollar investment. We'll circle back to that ethical tension later because it's a huge issue.
Okay, so let's say we get lucky. We have a druggable, validated, and economically viable target. We run our high-throughput screen. We get a hit. One of our million chemicals stick. Are we done?
Not even close. You're at the starting line. A hit is just a starting point. It binds, sure, but it might be incredibly toxic. It might not dissolve in water. It might be metabolized instantly by the liver and never reach its target.
So what happens to the...
Now, the medicinal chemists take over. This is the lead optimization phase. They take that initial molecule and they start tweaking it methodically. They add a methyl group here to protect it from being destroyed by the liver. They remove a hydroxyl group there to help it cross the cell membrane. It's an art and a science.
They're optimizing the ADME properties.
Exactly. Absorption, distribution, metabolism, and excretion. You are turning a crude hit into a refined lead compound. And crucially, at the same time, you are trying to create a molecule that is new, that is patentable as a unique composition of matter. It has to be novel.
Okay, so we've done it. We have our lead compound. It looks good in the computer models. It works in the test tube. Now we enter what you call Section 3, the preclinical gauntlet. Or as some people in the industry call it, the Valley of Death.
That's a good name for it. The objective here shifts completely. Up until now, we've been asking, does it work? Now, the primary overriding question becomes, is it safe? We are preparing to put this chemical into a living, breathing human being. We need to have some assurance that it's not going to kill them.
So this is where the animal testing comes in. Katzung lists a whole battery of tests. Acute toxicity, subacute, chronic, carcinogenicity, reproductive toxicity. It sounds exhaustive.
It has to be. You typically test in at least 2 species. One rodent, like a mouse or a rat, and one non-rodent, like a rabbit, a dog, or even a primate. You give them the drug every day for weeks, months, sometimes years.
What are you looking for?
Everything. You look at their blood work, you look at their organs under a microscope after the study, you're looking for signs of damage to the liver, the kidneys, the heart. You run separate long-term studies to see if the drug causes cancer. You run other studies to see if it affects fertility or causes birth defects. We need to see which organs, if any, light up with damage.
But going back to that translation problem, we just established that humans aren't mice. So how reliable is this really?
It's imperfect. That's the brutally honest answer. Goodman and Gilman is very clear that extrapolating from mouse to man is fraught with error. First, you have major metabolic differences. A rat's liver might have an enzyme that safely metabolizes the drug, while a human liver turns that same drug into a potent toxin.
So a drug could be safe in animals, but deadly in humans.
Yes, and the reverse is also true. We might toss out a potentially great drug because it happened to be toxic to the specific strain of rat we used, but it would have been perfectly safe in humans. We just don't know for sure until we try.
And what about the statistics, the numbers involved?
That's the other huge problem, statistical power. Let's say a serious side effect, like liver failure, happens in 1 in 10,000 people. If you only test 50 rats and 50 dogs, the odds of you seeing that rare event are practically 0. You will mathematically never catch it.
So rare but deadly side effects are essentially invisible in preclinical testing.
Correct. We are not looking for subtle or rare problems here. We are looking for the big, obvious toxicities. We are trying to filter out the outright poisons before they get anywhere near a person.
Goodman and Gilman makes a really important distinction between mechanism-based and off-target toxicity here. Can you explain that difference?
Absolutely. If a drug proves to be toxic in animals, we have to know why. Is it toxic because it's hitting the wrong target? That's an off-target effect. It's sloppy, it's binding to our intended receptor, but it's also binding to some other random protein in the liver and causing damage.
So if that's the case, the project might be salvageable.
Yes, maybe the medicinal chemist can go back to the drawing board, tweak the molecule's shape, and make it cleaner so it stops hitting that innocent bystander protein. But if the toxicity is mechanism-based, meaning it's toxic precisely because it's hitting the right target and doing the thing you want it to do, then the project is probably dead.
Can you give me an example of mechanism-based toxicity?
Sure. Think of a powerful anticoagulant, a blood thinner. Its mechanism is to stop blood from clotting. If you give too much, the patient bleeds uncontrollably. That isn't a side effect. It's the intended effect working too well. Or a strong immune suppressant for an autoimmune disease. If it works, it suppresses the immune system and the patient gets infections. You can't engineer that risk out of the drug. You can only manage the dose very, very carefully.
So you've survived the valley of death. You have a mountain of animal data. You think you're ready for human trials.
Yeah.
What is the pivotal legal step you have to take?
You file the IND, the Investigational New Drug Application. This is a massive dossier you submit to the FDA. It contains everything, all your animal safety data, your detailed manufacturing plans, your proposed protocols for the human trial.
And then you sit and wait for the FDA to give you approval.
Actually, it's a fascinating quirk of the system. It's a passive approval. The FDA has 30 days to review your IND. If they say nothing, you can start. If they see a major red flag, they'll issue a clinical hold and stop you. But silence is a green light.
So no news is good news.
In this case, yes. The default is, unless we stop you, go ahead.
Okay, so the 30 days pass, phone doesn't ring. We are now in Section 4. Clinical trials, this is the human experiment. This is famously divided into 3 phases. Walk us through Phase I.
Phase I is usually the very first time the drug enters a human body. The group is small, typically 20 to 100 people. And here is the crucial part. They're almost always healthy volunteers.
People who don't have the disease you're trying to treat.
Correct.
Wait, that seems counterintuitive.
Yeah.
If I have a new headache drug, why would I give it to someone who doesn't have a headache?
Because Phase I isn't about curing anything. The primary goal is safety and pharmacokinetics in humans. We want to know, how does the human body handle this drug? How quickly is it absorbed? Does it spike to dangerous levels in the blood? Does it cause nausea or dizziness?
And using healthy people gives you a clean baseline to measure that.
Exactly. If we give it to sick people, their underlying symptoms might confuse the picture and mask the drug's true side effects. We want a clean slate. We start with a tiny dose and escalate it very slowly in different groups of volunteers, watching like hawks for the first sign of any trouble.
But there's a major exception to that healthy volunteer rule, right?
A very important one, yes. If the drug is expected to be highly toxic by its very nature, think of a potent cancer chemotherapy or a new HIV drug, you cannot ethically give that to a healthy person. It would violate the core medical principle of do no harm.
You can't give a healthy college student a drug that's designed to kill rapidly dividing cells.
You absolutely cannot. In those cases, Phase I is done in patients who already have the disease and have often exhausted other options. For them, the potential benefit justifies the high inherent risk of the experimental drug.
So Phase I is all about safety. If it passes, we move to Phase II.
Phase II is proof of concept. Now, finally, we bring in people with the disease. It's a slightly larger group, maybe 100 to 200 patients. And now we ask the big question, does it actually work? Does it lower the blood pressure? Does it shrink the tumor? Does it reduce the number of seizures?
And this is where a lot of hearts are broken in the industry.
This is the highest rate of attrition in the entire process. This is where all those flawed assumptions from the mouse models come home to roost. You find out that the mechanism that shrank the tumor in the rat does absolutely nothing in the human, or you find that the dose required to have an effect is just too toxic for people to tolerate. Most drugs die in Phase II. It is the graveyard of good ideas and promising molecules.
But if it survives,
Yeah.
it shows a signal of efficacy. We move on to the main event, Phase III.
This is the gold standard, the randomized controlled trial, or RCT. Now you're talking about thousands of patients, often in multiple countries, and it has to be done under the strictest conditions, double-blind.
Meaning neither the doctor nor the patient knows who is getting the real drug and who is getting the placebo.
Correct. This is absolutely critical to eliminate bias. If a doctor knows a patient is getting the new wonder drug, They might subconsciously look for signs of improvement. If the patient knows, the placebo effect could be huge. Blinding removes all of that.
And this is the data that will make or break the company.
This is it. The results of phase 3 trials are what the FDA will scrutinize to decide on approval. These trials are incredibly complex, take years, and can cost hundreds of millions of dollars all on their own.
Goodman and Gilman brings up a really interesting and subtle point here about surrogate endpoints. What are those?
This is a really important trade-off between speed and certainty. Let's say you're inventing a new drug for heart disease. The ultimate hard outcome you want to prevent is death or a heart attack or a stroke. But to prove you prevented those, you'd have to run a trial for 10 years with 20,000 patients, just to wait for enough events to happen in the placebo group to show a statistical difference.
Which is incredibly slow and expensive.
Yeah. Prohibitively so in many cases. So instead, you use a surrogate endpoint, something you can measure easily and quickly that you believe stands in for the real disease outcome. For heart disease, the classic surrogate is LDL cholesterol.
So if the drug lowers cholesterol, you just assume it will prevent heart attacks down the line.
That is the assumption. And it's a very powerful one that has accelerated drug development. But it's also a dangerous one.
Why dangerous?
Because the assumption isn't always correct. There have been infamous cases of drugs that successfully lowered the surrogate. They made the numbers look beautiful, but didn't actually help patients live longer. Or worse, they actually caused harm through some other unforeseen mechanism. For example, certain antiarrhythmic drugs did a great job of cleaning up the ECG readout, the surrogate, but were later found to actually increase mortality.
They won the battle but lost the war.
A perfect way to put it. Relying on surrogates is a necessary shortcut, but it introduces a layer of uncertainty and risk that we have to acknowledge.
So let's say phase 3 is a success. The data looks good, even with the caveats about endpoints. You package it all up and file the NDA, the new drug application.
Right, and you also pay the FDA a fee. This is under the Prescription Drug User Fee Act, or PDUFA, which helps fund the FDA's review staff to ensure they can look at your application in a timely manner, usually within 6 to 10 months.
The FDA says yes. Champagne corks pop. The drug is on the market. But the story still isn't over.
No, that's a common misconception. Approved does not mean perfectly understood. We now enter phase 4, post-marketing surveillance. The experiment continues.
Why do we need this? If we've already tested it on thousands of people in phase 3, don't we know everything we need to know about its safety?
It's that numbers game again. Let's go back to our example of a rare but fatal side effect, like that liver reaction that happens in 1 in 10,000 people. In a big phase 3 trial of, say, 3,000 people, you will statistically probably see 0 cases. It's just not likely to show up. The drug's safety profile looks clean.
But once you launch it and it gets prescribed to 1 million people,
you now have 100 people with liver failure. Exactly. The real world is the ultimate clinical trial. We only see the low-frequency, but potentially devastating events when the population scales up from thousands to millions.
So how do we catch these things?
We have reporting systems like the FDA's MedWatch program. Doctors and even patients can voluntarily report suspected adverse reactions, and manufacturers are legally obligated to investigate and report any serious adverse events they learn about. This is how drugs sometimes get a black box warning added years after approval, or in rare cases, get pulled from the market entirely.
This brings us perfectly to Section 5, Regulation and History. You made a chilling statement earlier that regulations are written in blood.
It's the unfortunate reality of how the FDA came to be. It wasn't designed proactively in a conference room. It evolved reactively. It's a series of patches and amendments, each one put in place after a public health disaster. It's an archaeology of tragedy.
Let's walk through that timeline. It starts in 1906 with the Pure Food and Drug Act.
Right. That was the era of Upton Sinclair's The Jungle. That law was basically just about truth in labeling and preventing adulteration. You couldn't sell Dr. Bob's Miracle Snake Oil if it was actually just colored water and turpentine. You had to list the active ingredients.
But did you have to prove it was safe?
No! And you didn't have to prove it worked. You could sell a known poison as long as you labeled it correctly as poison. There was no gatekeeper for safety.
Until 1937. The elixir sulfanilamide tragedy. This is a story every medical student should know.
It's a foundational story. Sulfanilamide was a new wonder drug, one of the first effective antibiotics. A company wanted to sell it in a liquid form for children, which made sense. The problem was the solvent they used. To dissolve the drug, they used diethylene glycol, the primary ingredient in antifreeze. It has a sweet taste, but it metabolizes in the body into oxalic acid, which crystallizes in the kidneys and causes complete agonizing renal failure. Over 100 people, mostly children, died. And the terrible irony was that under the 1906 law, the company had broken no rules. They labeled it an elixir, and the only violation was a technical one because an elixir was supposed to contain alcohol, and this didn't.
So public outrage forced the government to step in.
It did. This led directly to the 1938 Federal Food, Drug, and Cosmetic Act. This was a landmark change. For the very first time, a company had to prove a drug was safe before it could be sold. They had to submit safety data to the FDA.
But not efficacy. You still didn't have to prove it actually worked.
Not yet. That took another, even more widespread tragedy, the one we alluded to earlier, 1962, thalidomide.
The sleeping pill.
Marketed across Europe and Canada as a completely safe sleeping pill and anti-nausea drug, specifically for pregnant women suffering from morning sickness. It led to an epidemic of phocomelia, a horrifying birth defect, where babies were born with limbs that were shortened or absent entirely, looking like seal flippers.
Thousands of babies were affected.
Yes. But crucially, not in the United States. The U.S. was largely spared because of one single FDA reviewer named Dr. Frances Kelsey. The company was putting immense pressure on her to approve the drug, but she kept dragging her feet. She was not satisfied with the safety data, particularly around nerve damage. She single-handedly held the line. She's a true hero of pharmacology.
And her skepticism saved countless American families from that fate. The public outcry led to what?
The 1962 Kefauver-Harris Amendments. This was the second landmark change. From then on, you not only had to prove a drug was safe, you had to prove efficacy. You had to submit data from well-controlled clinical trials showing it actually worked for its intended purpose. And of course, it mandated much stricter teratogenicity testing to check for birth defects.
It's so interesting, though, that Katsung points out a nuance about thalidomide today. It's not a banned substance.
No, it's an approved drug in the U.S., but under incredibly strict controls. It's used to treat complications of leprosy and certain cancers like multiple myeloma. It turns out its tragic flaw, its ability to stop blood vessel growth, is actually a therapeutic benefit in diseases driven by excess blood vessel growth. It proves that even a dangerous drug can be a powerful medicine if the risk is understood and managed. You just never, ever give it to a woman who could become pregnant.
I want to touch on the legal side of this. There's a Supreme Court case mentioned in Goodman and Gilman that's really important for clinicians to understand. Wyeth v. Levine. This defines the relationship between FDA approval and legal liability.
This is crucial. A patient, Diana Levine, lost her arm to gangrene because an anti-nausea drug, Phenergan, was injected improperly. It was given as an IV push, and some of it accidentally went into an artery, which is catastrophic.
And the drug's label warned against this, but didn't explicitly prohibit it.
Exactly. And the company's defense in court was, hey, the FDA approved our label, so we are immune from state-level lawsuits. The FDA's authority is supreme. It's a legal concept called preemption.
What did the Supreme Court say?
The Supreme Court said no. In a landmark ruling, they said that FDA approval is a floor, not a ceiling. It is the minimum standard for safety. It does not prevent a company from adding stronger warnings if they learn of new risks. And it absolutely does not shield a company from liability. This keeps constant pressure on manufacturers to be proactive about updating their safety labels.
That's a powerful check on the industry. And speaking of the industry, let's move to our final section, Section 6, Industry Realities. We have to talk about the money.
We do. As we said, it costs billions to bring a drug to market. The way companies recoup that massive investment is through patent protection.
A patent gives you 20 years of exclusivity to be the sole seller.
But here's the devastating catch for the industry. The 20-year clock starts ticking the moment you file the patent, which is usually way back in the invention or preclinical phase, when it's still just a promising chemical in a lab.
So by the time you spend 10 or 12 years navigating the preclinical gauntlet and all three phases of clinical trials,
your effective patent life is half gone. You might only have 8 or 10 years left on the market to make back your entire investment before generic competition comes in and the price plummets. This creates immense financial pressure to price drugs high and market them aggressively during that short window of exclusivity.
This pressure also leads to the phenomenon of Me Too drugs. Critics of the industry really hate these. Can you explain what they are?
A Me Too drug is a molecule that is structurally very similar to a blockbuster drug that's already on the market. For instance, after the first statin drug proved to be a massive success for lowering cholesterol, 5 or 6 other companies developed their own, slightly different statin molecules.
The criticism is that a waste of R&D resources? Shouldn't they be trying to cure Alzheimer's instead of making the sixth cholesterol pill?
That is the standard criticism, and it has some merit. It can seem redundant. But there is a defense for me too, drugs. First, simple competition tends to lower prices for the entire class of drugs. Second, and this is clinically important, patients are individuals. A patient might have terrible muscle pain on statin A, but do perfectly fine on statin B. Having those slightly different options is actually very valuable for tailoring therapy.
It's not a true class effect. There are individual differences.
Exactly. The subtle differences can matter a lot to an individual patient.
What about the diseases that get ignored entirely? The concept of orphan drugs.
This is the clearest example of market failure in medicine. If a disease is rare, affecting fewer than 200,000 people in the U.S., or if it primarily affects poor populations in the developing world, like many tropical parasites, the profit model simply doesn't work. A company can't make back its investment.
So what happens? Do those patients just get left behind?
Historically, yes. But now we have solutions. The government passed the Orphan Drug Act, which gives companies special incentives, tax breaks, extended exclusivity, to make it financially worthwhile to develop drugs for these rare diseases. And in the global health space, you have large philanthropic organizations like the Gates Foundation stepping in to fund research where the market won't.
The text also brings up the thorny ethical issue of global justice, specifically testing drugs in developing countries.
It's ethically very messy. On one hand, it can be much cheaper and faster to run clinical trials in developing nations. You have large populations of treatment-naive patients and less regulatory red tape. But is it fair to use a population as research subjects for a drug that they will never be able to afford once it's approved and sold in wealthy countries?
The Belmont Report Principles of Ethics would say, probably not.
Exactly. It can violate the principle of justice, which states that the group that bears the risks of research should also be able to enjoy its benefits.
Finally, let's touch on conflicts of interest. The FDA itself is funded, in part, by the very industry it regulates.
Yes, through the PDUFA user fees we mentioned. It's an arrangement that has successfully sped up review times, but it creates a perception problem. It puts the FDA in the awkward position of regulating the people who are essentially paying their electric bill.
And what about the missing data problem or publication bias?
This is a huge one. If a company runs 5 large trials for a new antidepressant and 2 of them are negative, meaning the drug failed to beat placebo and 3 are positive, they have a strong incentive to only publish the 3 positive ones. The doctor reading the journals sees a winning drug, a 3-for-3 success story. The reality is much murkier.
That is terrifying.
It is why clinicians need to practice evidence-based medicine with a critical eye. It's why we need to read these foundational texts. It's why we look for meta-analyses that try to include unpublished data and not just rely on the marketing brochures.
So we've tracked the entire journey, from molecule to mouse, from mouse to man, from man to the market, and we've seen the watchful, sometimes reactive eye of regulation hovering over it all.
It's a complex, imperfect system of checks and balances. It's flawed, but it's probably the best one we've been able to devise to balance innovation with public safety.
As we wrap up this deep dive, I want to leave you, the listeners, with a final provocative thought. We talked about placebo-controlled trials being the gold standard for proving efficacy. But Goodman and Gilman raises a serious ethical dilemma here.
It's the placebo paradox. The Declaration of Helsinki, which is the ethical guide for human research, states that it is unethical to give a patient a placebo if a known effective treatment for their condition already exists. You can't just let someone with a serious illness suffer and decline for the sake of clean data.
That seems right. You have to give them at least the current standard of care.
Yes, but if you do that, if you compare your new drug not to a sugar pill, but to the old effective drug in an active control trial, it becomes much, much harder to prove your new drug is better. You might need massive sample sizes, tens of thousands of patients, to show a small but meaningful improvement.
So we're stuck.
We're stuck. Do we prioritize scientific rigor, which often requires a placebo to see a clear effect? Or do we prioritize immediate patient welfare, which demands giving everyone at least the current best treatment? It's a fundamental tension at the heart of clinical research that doesn't have an easy answer.
Something to chew on.
Indeed.
Listeners, this was your conceptual map. We've given you the lay of the land. Now, it's time for you to walk the territory. Open Kadsung Chapter 1. Look at that diagram of the drug development timeline. Open Goodman and Gilman Chapter 1. Read the case study on Wyeth-Villavine for yourself.
These texts are dense, but they are the bedrock of our profession. Don't just memorize the drug names. Understand the journey, understand the trade-offs, understand the history.
Thanks for diving deep with us. We'll see you in the next episode.